Hello everyone, this is Amanda. The bacteria I talk about today is E. coli. So first of all, I want to share a narrative to you guys about the discovery of E. coli. So the bacterium was discovered by German Austrian pediatrician Theodor Astrick. He isolated a new bacterium from the feces of diarrhea patients, studied it carefully under a microscope, noticed its properties, and self-lastly named it E. coli colony. So Astrick noticed one very important property. It grew extremely fast under the laboratory condition. So by the time of Astrick dies in 1911, his finding had been used by multiple laboratories. And in 1919, the bacterium was renamed Astrea coli in his honor. So first, I would like to talk about the basic information of E. coli. So the scientific name of E. coli are Escherichia coli, and we abbreviate as E. coli. The culture requirement of E. coli our temperature are like 37 Celsius are the best temp to let the bacteria grows. And oxygen is not required, but if you present with oxygen, the bacteria can grow faster. For the pH, it's between pH 4.5 and pH 9. And this is the picture of E. coli. Classification of E. coli, the domain are bacteria, kingdom are bacteria, phylum are proteobacteria. The class are common proteobacteria, order is Enterobacterius, family are Enterobacteriaceae, genus are Escherichia, and species are Escherichia coli. And this is the image of E. coli under the light microscope. The actual size of E. coli, this is a picture of electron micrograph of E. coli, and the radius is about 0.5 micrometers, and the diameter is 1.0 to 2.0 micrometers long. So for the life cycle, E. coli reproduced by two means, which are self-division and the transfer of genetic material through a sex peleus. So in most bacteria, conjunction depends on fertility factor that is present in donor cells and absent in the receive cells. Cells that contain F, which is the fertility factors, are referred as S, and cell lacking F or F. E. coli reproduced by binaural fashions, and the E. coli use the AGT through the mobile genetic elements to increase the genetic variation. For the sex peleus, cells containing F produce the sex pele, which makes contact with a receptor on F cells and pulls the two cells together. So the DNA is transferred from F cell to the F cell. Conjunction can take place only between a cell that processes F and a cell that lacks F. And this is a flow chart of E. coli as a whole cell, how they attach, invasion, these kind of things. So for the features of E. coli is a gram-negative, road-shaped, and no spore forming. The mortality of E. coli are swimming and swarming. The flagella arrangement, like they always arrange lateral around the body. And here's a picture how they arrange between the bacteria. The medium used for E. coli is Gram-Macaque agar. So the disease caused by E. coli, urinary tract infection, respiratory illness, diarrhea, all these diseases are the common bacteria will be caused by E. coli infection. The main reservoir of E. coli strain is grass-feeding animals and cotton in particular. Glucose 6-phosphate, fructose, all these things are major intermediates, and eating contaminated food or drinking contaminated water are the reason frequently cause the disease. And people who are most at risk are adults age 65 and older, children younger than 5 years old age, and people with weakened immune system, and including the pregnant woman. So the symptom prevent and treatment. So the symptom of E. coli infection. So first of all, they will have diarrhea, which can be bloody, and most of them have stomach cramps. And third one, some also have vomiting. How we prevent it? So we can wash our hands through like soaps and running water. Second, we can follow the 4 steps to the food safety when prepare food, clean, separate, cooking, and chill. Third, we can use a food thermometer to make sure the food has reached a safe minimum cooking temperature. So the last one, don't drink untreated water or swallow water. All these tips are come from the CDC, the Center of Disease Control. So the treatment of E. coli infection. So first of all, we need to make a diagnosis. So doctor will send a sample of your stool to a laboratory to test for the presence of E. coli bacteria to diagnosis of this illness. And then the doctor will tell you to rest and foods to help prevent dehydration and fatigue. There's no any other ways can treat the E. coli right now. That's it. Thank you so much for you guys watching.